Okay, so I'm going to be focusing on just a brief introduction to the background of CML and the development of the international scale. And Helen's going to focus on the issue of standardization, basically our experience over several years, and particularly focusing on the lessons learned, because some things went well, some things I think we would do differently if we could go back in time. So then, CML, for those of you who are not familiar with it, is basically the paradigm for personalized medicine. Hang on, I'm just going to shut the window. Some building outside. CML is defined, all CML patients have the BCR ABL fusion gene, but going back in history, in 1960, this small derivative chromosome here, which was not recognized, the nature of it was not recognized immediately, but this was the first consistent chromosome abnormality associated with any human malignancy and identified in a series of patients with CML. It took chromosome banding techniques, which were developed about 10 years later, to understand that the Philadelphia chromosome was the smaller derivative of a reciprocal translocation between chromosomes 9 and 22, and work over the following 10 years or so to understand that this disrupted two genes, ABL on chromosome 9, BCR on chromosome 22, producing the BCR ABL fusion on the Philadelphia chromosome. Parallel biochemical analysis showed that ABL was a new class of enzyme, a tyrosine kinase. So it's a critical signaling protein that phosphorylates tyrosine residues. And work in mice demonstrated that BCR ABL as a sole abnormality was capable of giving rise to CML. So this served as the backdrop for the development of targeted therapy in the form of ATP mimetics. So normal BCR ABL binds ATP, and that's the source of the phosphorylation reaction. Compounds such as imatinib bind this ATP binding pocket, but are inert, and so basically switch off signaling. And the clinical results have been absolutely transformative. So a disease which was almost invariably fatal. CML patients now have a life expectancy which is getting close to the normal age-matched population. So why is it important to monitor molecular response to tyrosine kinase inhibitor treatment in CML? Well, there's abundant evidence that the degree to which the bulk of disease is reduced is the most important prognostic indicator for any individual. And these measurements trump all upfront risk scores. And these have been translated over the years into national and international guidelines, which specify time-dependent treatment milestones to define whether a patient is responding optimally or suboptimally and may need a change of treatment. And in recent years, we have this remarkable finding that patients who achieve sustained deep molecular response, about half of them maintain remission when they stop treatment. So they're operationally cured. So just to focus on the BCR ABL fusion itself. So by definition, all CML patients are BCR ABL positive. At the level of genomic DNA, the genomic breakpoints are very widely dispersed, particularly within the ABL gene. You can design patient-specific DNA PCR assays, but that's an expensive approach for routine monitoring. But after splicing of the primary transcript, the great majority of CML patients, so 98%, express one or both of these two transcript types, which differ by the presence or absence of a single BCR exon. So these are known as E13A2 fusion of BCR exon 13 to ABL exon 2, or E14A2. And there are different assay configurations used internationally, but the most commonly used uses a single set of primers and a probe, which will amplify both of these fusions together. So E13A2, A14A2 are very convenient targets for either qPCR or digital PCR or other techniques. It's important to remember there are these 2% of CML cases that express a whole range of alternative BCR ABL fusions, and that definitely complicates the picture. But by and large, the focus is on the 98% of patients expressing these two transcripts. So the process is deceptively simple. We start with peripheral blood. So definitely in CML, blood and marrow are equivalent, provided that you use red cell lysis, not lymphoprep. RNA is extracted, converted to cDNA, and then there are two measurements. Most labs use qPCR still. Digital PCR is a perfectly good alternative, both for BCR able to estimate an estimate of the number of leukemia cells, basically, and then for a reference gene to estimate the quality of the sample. And that quality is often highly variable. Reverse transcription is a highly variable process. We're getting samples, clinical samples may reach the lab in a couple of hours. They may take 2 days or more. Fundamentally, with RT-PCR, we're dealing with a difficult situation. If you're using a DNA-based assay, you have a natural unit of measurement, which you can relate to the number of cells, if you assume that there's one rearrangement, for example, per cell. With RT-PCR, we can't do that. We need the reference gene as a critical parameter to relate to. And one of the big problems we've had in CML is that there's no universally agreed RT-PCR reference gene that fulfills all the various requirements. Most labs internationally use ABL as a reference gene, the normal ABL gene. There are many labs that use GusB or BCR. And this is a big complication because fundamentally these are different units of measurement. But the raw values we derive, so for positive specimens where BCR-ABL is detected, we're looking for the ratio of BCR-ABL to the reference gene. For samples where BCR-ABL is not detected, the number of reference gene transcripts indicates how good that sample is, and so the degree to which you can exclude measurable residual disease. So this was all fine. Labs, a number of labs set up MRD tests. These are research labs largely. The clinical question then was about relapse after bone marrow transplantation. But the whole world in CML was transformed by the development of tyrosine kinase inhibitors. And the real focus in terms of standardization was on the IRIS clinical trial in 2003, which formally demonstrated the superiority of imatinib over conventional treatment. And the primary endpoint of the trial was cytogenetic response, but there was a bolt-on investigational study to look at the value of molecular monitoring by RT-qPCR. And there were 3 labs, one in London, one in Adelaide, and one in Seattle that performed the monitoring. These assays were not standardized. So historically, the standardization arguments focused initially around contamination, subsequently around which is the best reference gene, and that was really never resolved. So these 3 labs, 2 used ABL, 1 used BCR, and these tests were not standardized. And that became apparent when you look at these results here. So these are not identical samples compared between labs, but these are a large number of samples compared at identical time points. So you'd expect them to largely overlap. And you can see the yellow lab here was a particular outlier, but even the green and red labs here were not ideally close. So this was a bit of a crisis for this study. So they took really an arbitrary decision. What they did is they shared 30 chronic phase CML samples, so pre-treatment samples. They measured them in each of these labs using BCR as a reference gene and used that as a normalization factor. So then all subsequent results were related to this standardized baseline. So for example, the value corresponding to what's called a major molecular response, which is a 3-log reduction, it's from that standardized baseline, not the pre-treatment levels for each patient. So in all other contexts that I'm aware of, when you're talking about minimal residual disease, you're measuring the level of disease pre-treatment and then on treatment and looking at a log reduction. And this basically worked for the trial, but it had very important follow-on consequences because this was used basically to define the international scale. So the international scale was made possible by the fact that the Adelaide lab, one of the IRIS labs, had excellent traceability data going back over a period of years. So we could basically relate results to the Adelaide lab. So we have our local assays using different reference genes. Even for individual reference genes, people use different primers and probes, and they can have important consequences. Different bits of kit, qPCR or digital PCR. And the concept is that by using calibrators or conversion factors, that these local assays can be converted to the international scale, expressed as a percentage, with this value of 100% corresponding to the IRIS standardized baseline. 0.1% major molecular response is a 3 log reduction from that baseline. And deep molecular responses are basically 4 logs or greater from that standardized baseline. And when this was proposed, everyone thought it was great because it accommodated people's favorite assays, basically different reference genes. But implementing this internationally has been a real challenge. So just some thoughts about if we could go back in time and do the IRIS trial again, or the molecular component of the IRIS trial. So I think having a small number of labs doing the monitoring was good. 1 is probably too few, 2 to 3 is probably about right. If we could have possibly have agreed on an identical assay platform and reference gene, that would have certainly made things a lot simpler going forward. We'd like to have had defined criteria for the derivation of results. Very interesting if you present, or we've showed in the past, if you presented labs with identical data, they would derive different results depending on how they treated duplicates, triplicates, etc. So you need to define criteria, cross-validation of results between centers. And I think very importantly, we would have wanted to measure levels of disease at diagnosis or pre-treatment and then after treatment, so we could look at a patient-specific log reduction. I think it's important to understand that the international scale for BCR-ABL measurement very much came about because we couldn't agree on what was the best reference gene, and because it was not standard practice to measure levels of disease pre-treatment. So, as I said, implementing the international scale has been a huge challenge. What was basically the gold standard technique was developed by Sue Branford in Tim Hughes's lab in Adelaide, and it was a process of patient sample exchange. So basically, there was an initial exchange of samples. These were patient lysates. They could be extracted, put through the whole process, reverse transcribed, qPCR. So here, the reference lab, the initial reference lab is the Adelaide lab. Lab 5 here is a participating lab. And so these 16 samples you see, they're all displaced off the line of identity, but it's a simple mathematical transformation to bring them up to the line of identity. So that's then the conversion factor. And then there's a subsequent follow-up with additional patient samples to confirm that basically the assay is stable, that conversion factor has worked. So you can see these 33 samples are all nicely dispersed along the line of identity. And then there'll be a revalidation process at regular intervals. So this was great. It worked well for most labs, but it clearly wasn't scalable. And we rapidly ran into big problems. We had a very large program in Europe, the EUTOS program, that promulgated conversion factors, but it was a huge amount of work. So there was a clear need for a more robust approach, including the development of generally available validated reference reagents. And I'm going to hand over at this point to Helen, who's going to talk about what we did and what went well and what didn't.
Okay. So over the last sort of 16 years or so, Nick and I have been working on the establishment of some higher order reference material and also secondary materials for CML testing, largely by detection of the BCR-ABL transcript by real-time PCR. So the first material that we made is a higher order material. So it was the first World Health Organization standard for BCR-ABL. And this is actually a freeze-dried mix of HL60s and K562s. And there's a panel of 4 freeze-dried cell samples ranging from about 10% to 0.01% on the international scale. And they have certified values for labs using ABL, BCR or GUS as a control gene. And then moving on later in the, in 2015, we looked at making a certified plasmid reference material. So this is a kind of critical component of the RQ-PCR assay, where labs use a plasmid standard curve to read off their ABL and BCR-ABL transcript numbers. So we work with the IRMM, the Institute of Reference Materials and Methods, to produce an E14A2 plasmid. And this plasmid has the target, so the BCR-ABL transcript, ABL, BCR and GUS clones in single copies onto the same plasmid backbone. So each, so a value of 10 for ABL should give a value of 10 for GUS, BCR and BCR ABL. This is available commercially from the IRMM or from Sigma, and is actually a European reference materials. This is it is a higher order material, but not quite as high order as, say, something like a CE marked material. And then fairly recently, Novartis drove the production of a secondary material. So they basically replicated the World Health Organization material, calibrated that to the World Health Organization material, and that was then further commercialized by Thermo Scientific. So Acrometrics is a subsidiary of Thermo Scientific. And this went a little bit further than the BCR ABL WHO material in the fact that they added a very low level standard, so the 0.0032%, EMR 4.5. Currently, though, that's only available for research use only. So we learned quite a lot of things during the 16 years. I think the first one is that it's not necessarily a simple process to make a reference material. It's not just a case of mixing two cell lines together and getting somebody to stamp the bottle for you. So I would say one of the things to be aware of is don't be tempted to just start making a reference material before you know the route you're using to establish it as a higher order material or a secondary material. There are lots of different routes. So for example, our first material was a World Health Organization certified material. We collaborated with the NIBSC. They're very experienced at producing WHO material. There are some caveats for it to be a WHO material. It needs to be applicable to countries worldwide. So it's not just first world nations doing kind of NGS testing, and the field trials need to be very international. Another route is to CE mark or ERM certify the material. So we worked with the IRMM on this. They're very experienced at ERM plasmid-based material, mainly sort of GMO-based detection. And NIBS have also now CE marked some of their genomic reference materials. Obviously, with Brexit, the CE mark kind of isn't technically recognised in the UK, so you need a UKCA mark, but I believe that they are relatively similar in terms of what you need for production. For FDA approval, we haven't had any experience with this, although we are hoping that Acrometrics will take their panel through either FDA approval or CE marking. Obviously, some of this production is outside of the scope of kind of a clinical lab. So I think conversations with the reference material providers such as NIBS, NML, RMM are really helpful to choose the right or the most straightforward certification for each different type of material. And probably a word of warning, it can be a very bureaucratic process. So that leads on to lesson 2, which is don't underestimate the time that it will take to produce a primary reference material. It will probably take longer than you think. This timeline just shows the stages of the production of the WHO material. So back in 2006, John Goldman approached Nick about the possibility of what at the time was the National Genetics Reference Lab in Wessex to help produce a BCR-able reference material. There was a huge need. People were doing very complicated sample exchanges to derive conversion factors. So over the next couple of years, we looked at different types of formulations, what cells you should mix together, what should you be using, blood combs, KG1s, HL60s. Admixing cells isn't necessarily a straightforward process. We then worked with NIBS to trial various freeze-dry protocols, did a small field trial, and then there are things that you have to incorporate, such as stability testing, accelerated degradation testing. And then in 2008, we were happy that we had a formulation that we could use. But then we had to finalise culturing of the cells, booking of a freeze-dry facility. We had to book that 9 months in advance to a specific day. And then we had contracted the European cell collections at Portland to grow 40 litres of HL60s and 4 litres of K562s. And then we kind of had a D-Day of the 3rd of September, where we were admitted to ECAC to work on site, make the dilutions, take them up to NIBSC, and then the day of the freeze dry, basically it was the first time that 4 different materials had been filled on the same day. So it's quite a complex procedure, and we ended up making 3,500 vials approximately of each material. And that kind of wasn't the end at that point. Then that material needed to be field trialed. It had to have microbiology testing, homogeneity testing, accelerated degradation, field trials to assign IS values, and then submission of all that data to the WHO for approval. So that was finally signed off in November 2009. And then in March 2010, it was released for use. Because of the limited quantity, that material was only available for commercial labs who are calibrating secondary standards. So this isn't available to clinical testing labs to derive their own conversion factors. So lesson 3 really is think really carefully about the composition of the reference material before you commit any time and money. If we could go back again, I think with the WHO material, we used far too many cells per vial. So this pushed the control gene number up. At the time, it was higher than most labs are achieving. It's probably comparable at the moment. And then the IRMM plasmid, currently the lowest standard is about 10 copies per microliter. And if you think labs tend to use 5 microliters per assay, that bulks up the bottom copy to 50 copies, and ideally labs now are detecting kind of 3 copies. The IRMM were really reluctant to produce a standard below this because of the problem with the accuracies surrounding quantification, but the labs needed that to quantify, but the IRM needed the accuracy to certify the material. So we kind of got into this chicken and egg situation where what was required was below what you could accurately quantify. So there are several considerations. What's appropriate? Do you want a cell line, patient material, synthetic material, bioinformatic material? Choice of cell lines isn't necessarily straightforward. You need to think closely, especially for qPCR. Are your control gene ratios sensible and in line with patient material? How many cells you'd need per vial? What's your kind of optimal RNA yield, target and control gene numbers? That has to be kind of biologically sensible. For a low-level sample, how low do you need to go, and how are you going to quantify that? Assessing whether accurate measurement is actually needed, acceptability criteria, and also production of negative control material. This can be surprisingly difficult. Sometimes you can get sort of non-specific amplification events which make accreditation of that difficult. The 4th lesson is around field trials. These are an essential part of the process and will differ depending on what route you use. But mainly it's sending material out to a wide range of labs and checking that it does what you want it to do. And I think a real word of warning here, which Nibs kind of drummed into me early on, was however clear you think your protocols are, someone will always do something different. So really get as many people to sense check these as you can. We've sent samples out to kind of 80 plus labs year upon year, and you still get people doing something a bit odd. Hints like, if you're asking people to send files back to you, ask them to add their name to the file name, otherwise you end up with 50 files all called the same thing. So some kind of online reporting system like NEQAS use is very preferable. And be careful to clearly define the aims of the trial. So what are you assessing? Are you wanting data and quantification? Is it the differences in technology, commutability? you don't want to get data back and then realize that you forgot to collect the essential bit of information. So if in doubt, ask for more information than you think you're going to need. And then the choice of labs, are you just wanting to test it with expert labs or all labs, local, European, worldwide? And we still have labs that experience issues using lyophilized materials, so there is a need there for education. And in our last round, we did actually send out a video showing people how to use the materials. So I think something that's probably important for the AML materials would be future-proofing, both in terms of levels of detection, but also the new technologies. So, for example, the WHO material, the secondary standards incorporate MR 4.5. So when we started, the focus point was MMR. And obviously now people are looking at deeper molecular responses. And in the IRMM plasmid, we did survey labs to find out what was a sensible region to be cloned. But now some of the newer assays, for example, the BioRad digital PCR assay doesn't amplify the ABL target from that plasmid. So there are a lot of considerations. So what's your dynamic range? How will a material cope with transfer from qPCR to NGS? So a lot of cell lines will be perfectly acceptable for RT-PCR, but when you move into NGS, they are going to have complex karyotypes. So if people are looking at whole genome, that will affect analysis. And also the different types of NGS, Illumina, they're shorter targets, captures, amplifications. Nanopore, you might be looking at more whole genome type analysis. So, cell line material may not be so okay for NGS applications. Also, making composite controls, if you want to sort of try and be clever and get all of your mutational profile within one material, then that will inevitably it's going to need a lot of developmental work to check that will be compatible with the newer technologies. And then maybe just a thought, would kind of bioinformatic controls be useful instead of or in addition to sending out variant call files, for example? So lessons learned, 6 is around kind of batch sizes and maybe a word about commercialization. So the WHO material inevitably was a limited resource. It was very cell culture intensive. So that's a restricted material and its usage is restricted to commercial companies making secondary materials. So it's not available directly to labs. With the IRMM material, obviously it's a plasmid, it's very easy to produce in large quantities. It was relatively simple assigning copy numbers using digital PCR, and it's supplied commercially, although there have been a few problems with delays to shipments and lead times. With the Acrometrics panel, unfortunately, that hasn't been calibrated for labs using GUS-B or BCR. So I think it's a word of warning that once you commercialize something, you tend to lose control of future developments. You can suggest things, but if it's not commercially favorable, then that might not happen, which is understandable. So lessons learned here are really clearly specify what you expect in contracts before you begin. Assess the predicted usage. And we think commercialization is hugely beneficial, but just be aware of you can lose control over formulation and future developments, and be careful to make enough primary material because that's going to be one of the nightmares to go back and redo, so make a big enough batch. So really the key to all of this is collaborations and funding. So firstly, you need to define the requirements with the scientific communities. There's no point in making something that's not needed or people won't use. Start conversations really early with reference material producers. It requires a lot of collaboration and a lot of funding. Field trials generally work on kind of goodwill from testing labs, and I think now things are getting economically tighter. It's very difficult to ask labs to do large scale field trials as it's taking up staff time and resources. and also make sure that those conversations are actually turn into production of a useful material. It really was a struggle over the last 16 years to get material out there. And also industrial partners may be helpful if the material has a potential for commercialization. One of the areas that still is kind of largely unaddressed is production of sort of day-to-day controls, things for rare samples. It's really difficult to get these made in a reliable way that isn't really labour and cost draining. So, for example, high and low level lysate controls that people can use on a day-to-day basis. At the moment, there's no kind of mechanism for sending those out in a standardised way. Negative control samples are generally lacking, and they can be quite hard to make. We've made plasmids for atypical fusions, and we are in the process of banking them with AdGenes so that people can access them on their own kind of risk. And we know that many labs have useful bank material that could be used for controls but can't find routes to production. So it'd be nice to hear from some of the reference material providers about maybe how that could be facilitated. And lastly, the control material have been used successfully. So this is a slide of the EUTOS labs, the validation of their conversion factors. So the bright green is where the validation is optimal, green is satisfactory, and orange is sort of unvalidated, or it didn't really validate on repeat testing. So in 2016, everybody was kind of, there was like a 30-30-30 split. And by the end of the study in 2021, over 90% of the labs were either optimal or satisfactory. So these are just some jottings that I did when Stu sent me the sort of remit for the meeting. Initial thoughts, AML testing, it's much more complex than CML, but you do have the advantage that you're not dealing with the international scale, which is kind of Pandora's box. It's really important to identify potential sources of funding and collaborative partners early on. Scope some willing field trial participants, prioritise your materials. My temptation would be to start with plasmid-based materials. It's very easy to make and produce. There are routes available, and they also give a lot of information to labs, particularly if your control gene and target on the same plasmid. You can look at relative efficiencies, absolute efficiencies, monitor CTs in every run, and they're easy to make. The cell-based materials, I think, need quite a lot of developmental time and need to be tested with an eye to moving to NGS, and possibly bioinformatic material might be another possibility. So obviously all of this couldn't have been achieved without extensive network collaborators. So NRBSC, IRMM, Novartis, Thermo Fisher. This all started way back in kind of 2002 with certified Crumgen, certified reference materials and genetic testing with David Barton and Simon Patton, and kind of grew out from there. And there are hundreds of labs that we've worked with over the years.